Hello and welcome to Sam Biology class. Friends, gas liquid chromatography GLC is a form of column chromatography. Its principles are similar to those of high performance liquid chromatography, but the apparatus is significantly different. The stationary phase of GLC consists of a high boiling point liquid material such as silicon grease or wax. The stationary phase is either coated onto the internal wall of the column or supported on a granular inert solid packed into the column. The mobile phase is a gas. The use of GLC is confined to analytes that are volatile but thermally stable. In this technique, a gas stream, also known as the carrier gas, flows continuously through a column at a flow rate that is controlled. When a small amount of sample is introduced into the gas stream, different analytes present in the sample are carried into the column. Inside the column, different analytes undergo repeated partition between the stationary liquid phase and the mobile gas phase. The analyte which is most volatile favors the mobile phase and hence fast elute out of the column. Other analytes which are less volatile are eluted later on. Gas liquid chromatography thus exploits differences in the partition coefficients of volatilized analytes between a stationary liquid phase and the mobile gas phase. The analyte which is most volatile will interact strongly with the mobile phase and hence it will take very less time to come out of the column. Other analytes which are less volatile will interact strongly with the stationary phase and hence they will take more time to be eluted out. The partition coefficients of analytes are inversely proportional to the volatility of different compounds. This figure shows the separation of three different analytes A, B and C. Out of these, analyte A is most volatile. Because of this, it interacts strongly with the mobile phase, and because of this interaction, analyte A moves faster through the column and takes less time to be eluted out. The second analyte B is moderately volatile, whose interaction with the mobile phase is less compared to that of analyte A. And because of less interaction with the mobile phase compared to A, analyte B takes more time to come out of the column. And the third analyte C is least volatile. Because of this, its interaction with the mobile phase is least, and as a result, at last, it is eluted out. After separation, the individual analyte reach a detector which reads their concentration and converts into an equivalent electrical signal. After amplification, the signal is measured by a continuous recorder. When the magnitude of the signal is plotted against time required for different analytes to be eluted out, a pattern is obtained. This pattern is called a chromatogram, which is made of a number of peaks. Each peak corresponds to one analyte. The characteristic positions of each peak is measured in terms of the volume of gas that has traversed through the column between time when The sample was applied into the column and the time when the analytes are eluted out. This volume is called the retention volume, which is fundamental to the gas-liquid chromatography. The retention volume is used for analysis of the sample. Like other chromatography, the area under each peak is usually proportional to the concentration of the analyte. This figure shows the major components of a gas-liquid chromatography system. The components are a carrier gas supply, a sample injection point, a long column which has been coiled, the heated oven, a detector, the amplifier, and a continuous recorder. In this technique, the temperature of the column is raised to 50 to 300 degrees centigrade to facilitate the volatilization of the sample. Gas-liquid chromatography is routinely used for the analysis of low polarity compounds because of its high sensitivity, reproducibility, and the speed of resolution. Analytically, it becomes a more powerful technique when coupled to mass spectrometry. It has several applications such as the separation of components of tobacco smoke, organic acids, atmospheric pollutants, essential oil, volatile vegetable oil, and different solvents. It has also been used to study the thermodynamic properties of different solutions. GLC is used to study the reaction rate, energies, and the mechanism. Finally, vapor pressure, heat of vaporization, molecular weight, bond angle deformation, ionization potential, and electron affinity has also been analyzed by this technique. Friends, this is all about the principle and applications of gas-liquid chromatography. For more updates, subscribe my YouTube channel, Sam Biology Class.